I will now move on to the next presentation. So I'd like to introduce Will to present T-Cell Tool Spotlight, TCRMatch. Thank you very much, Will.
Thanks, Nina. I'll share my screen and get started. It's good to be with all of you today. So yeah, we're going to be talking about TCRMatch, which is a tool we've been developing over the past about a year now. And if you're working with TCR repertoire sequencing data, this may be of interest to you. I'll start by just reiterating that this tool is not yet live. We are still working on the manuscript, and we want to get that taken care of before we make the tool available to the public. But when it is available, it'll be available on the IDB analysis resource. It'll be on VDJ Server, which is another site run by some collaborators of ours. And it'll also be available as a standalone download, so you can run it on your own machine. I'll start with some background. So we've already covered a lot of this before, and many of you are already well aware of it, but we know that MHC molecules present antigens that are recognized by T cells, specifically T cell receptors. And this is what we chose to focus on. In particular, we chose to focus on the interaction of the TCR and the antigen or epitope for the purposes of this tool. So to go into a little more detail about the TCR, it's typically a dimeric protein complex comprised of an alpha chain and a beta chain. And these come from the TCR beta and TCR alpha gene loci, which are assembled by this fascinating VDJ recombination that generates the vast diversity of TCRs that we have in our immune system. And I won't go into much detail on how that works, but suffice it to say that there's essentially a random shuffling of V, D, J, as well as C segments of genes that generate the incredible diversity that we have of TCRs in the immune system. And within these, each of these chains, there are 3 complementarity determining regions called CDR1, 2, and 3, which, as you can guess from the name, helps confer the specificity of the TCR for the epitope, as well as the MHC complex, which is presenting the epitope. So repertoire sequencing has become a really popular technique in modern times now that we have the capability to sequence massive numbers of cells. Generally, there's several different ways that this is done, but generally you'll always get sequences back that include the CDR3 beta region. And the reason this is such an important region to sequence is that this is the part of the TCR that interacts directly with the epitope, and it's also the most variable. So when the gene segment shuffling of VDJ recombination is occurring, you can see that the CDR3 beta region, I'll turn my laser pointer, stretches from the V segment to the J segment. And so all of the diversity of these gene segments is encompassed by the CDR3 beta region. So when you sequence TCRs this way, you'll get results back that look like this. Usually the CDR3 beta is around 10 to 12 amino acids in length. It's flanked by a cysteine and a phenylalanine. And sometimes those residues are included in the CDR3, but officially, according to the IMGT conventions for how CDR3 is defined, those residues are excluded. Despite all of this, TCR sequencing doesn't reveal the epitope that's actually recognized by the CDR3 beta, which is an important point. So you can run other experiments that help figure out what the epitope is in parallel to sequencing TCRs or prior to sequencing TCRs. But the sequencing itself will not tell you what the epitope is that the CDR3 recognizes. On the other hand, we do have a lot of data about CDR3 beta sequences and epitopes, and it's all in the IDB, and we can use that data to make educated epitope predictions for CDR3 beta sequences. So the goal for this TCRMatch tool was to build a tool that can search input CDR3 betas, such as from a repertoire sequencing experiment, against the IDB for similar sequences or matches that have a known epitope specificity. And the underlying principle here, of course, is that if two CDR3 betas are similar, then there's a good chance that they recognize the same epitope. And we know that from prior research. So the data set that we used was about 24,000 CDR3 beta sequences, as well as their corresponding epitopes from the IDB. And we tested 6 similarity algorithms, 4 of which were based on aligning one CDR3 against another, one of which was based on the Levenstein distance or edit distance between 2 CDR3s. And that's basically just the number of insertions, deletions, the substitutions that are required to transform one sequence into another. And finally, the algorithm that we then dubbed TCRMatch, but which had previously been described and implemented in these 2 papers. So that's the one that I'm going to discuss today, because, spoiler alert, it turned out to work out better than the other 5. So that's why I'm going to talk about it. So the TCRMatch algorithm relies on a modified BLOSUM62 substitution matrix to calculate similarity scores between sequences. And so if you don't know what this matrix is, basically it's a map, or it's a matrix, a grid of, if you replace the amino acid on the left with the amino acid on the top, is that a favorable or unfavorable substitution? And so you can look at examples like if you switch an isoleucine, an I, for a leucine, an L, that gives you a score of 2, which makes sense because those amino acids are very similar. On the other hand, there are other examples where if you switch an isoleucine for a glycine, you get a score of -4, because those are not as interchangeable. And so this matrix is a big part of how TCRMatch works. So I'll just quickly go through what it does. So basically, it compares 2 sequences using all possible k-mers. So if we start with k equal to 1, if we're looking at one residue at a time, we'll compare the first A in this sequence to the first A in this sequence, again referring to BLOSUM62 to see how favorable that lack of substitution in this case is. And then we move on to the second residue in the second sequence. We check how that compares. Then we compare the A to S, A to I, and so on for k equals 1. And then we move on to k equals 2. So we are now looking at dimers, but we're still looking at one amino acid at a time. So within the first dimer of each sequence, we look at A to A, then S to S, and then we shift over to the next one in the second sequence, A to S, S to S, A to S, S to I, A to I, S to R, and so on for k equals 2. And we do that for the rest of the possible k-mers up to k equals 10, which is the length of this shorter sequence. And ultimately, we compute a score between 0 and 1. If the score is 1, it means it's a perfect match. You're comparing the exact same sequence. 0 would be the opposite of a perfect match, if such a thing is even possible. I think the lowest scores we typically see are around 0.3 or 0.4. But yeah, so evaluating the performance of TCRMatch, we did that by following, by basically seeing how well we did in testing the IDB data against itself. We wanted to see if we recovered the same epitopes in the matches that were recognized by the input sequence. So we start by getting a score with every possible pair of sequences. And so this is obviously a simplified version where we're testing 1 against 4. And let's say that we impose a threshold for what we call a match of 0.9. In this case, we will eliminate the 4th candidate match here, leaving us with 3 candidate matches that score above 0.9. And then we can look at the epitopes that are recognized by each one. So we can see that epitope 1 is recognized by the input sequence as well as by the 1st and 3rd match. But epitopes 2 and 3 are not recognized by any of these 3 matches. And 3 matches also recognize epitopes that are not recognized by the input sequence. And so all of that information gets tallied up in our precision and recall statistics. And we do this at a variety of thresholds for each method. And the output looked like this. So TCRMatch we found was the best performing, especially in this early part of in this left half of the graph. And so this was good to see that we were able to get precision above 50%, recall for in the neighborhood of 0.2, and TCRMatch just in general, it outperforms the other methods. And then eventually everything converges as we get into more and more lenient thresholds. We're getting worse and worse matches, and as a result, we lose precision while gaining recall. The dashed line refers to the average performance for a randomized data set, and that is a roughly 0.08 in precision. So if you were just to pick a match at random and see if the epitopes matched up, you'd have about a 0.8 chance. And that's actually surprisingly good, but that's just reflective of the unfortunate bias that we have in the epitopes that have been studied within the field for TCRs and epitope pairs. So as time goes on, we can expect a greater number of epitopes to be studied, and hopefully we'll get a more representative data set of TCRs and epitopes. We also tested this on an independent data set published by 10x Genomics, just to make sure that it wasn't a fluke by looking at the IDB data. And once again, we saw TCRMatch outperforming the other method we tested, which was Levenstein distance. So now I'm going to actually show you what the tool looks like. And so hopefully this will all run smoothly. If you're not seeing the new Google Chrome page, then please let me know. But this is the TCRMatch web server. And so like all the other tools, it comes with a help page, which will explain everything that needs to be filled out. It comes with example data. I'm going to run this second one. And so you can enter your sequences here. You can also upload a file if that's easier. We recommend that people keep this trim sequences option on because, as I mentioned before, often these CDR3 sequences do have the C and the F included at either end of the sequence. But we, in accordance with the guidelines from IMGT, we prefer to trim those off and before aligning it, before trying to find similarity.
Sorry, you can 5 minutes left as well.
Okay, great. So you can select a filtering level as well. There are a few options here. We recommend the highest setting of 0.97, which will give you the most accurate results. It might not give you a lot of results, but you can have pretty good confidence that you're getting good results. Meanwhile, there's a person with a leaf blower outside my window. So hopefully you can still hear me. There are lower settings as well if you want to get more results, and you can even show all the matches if you want every single TCR match score for your input sequences against the IDB. And you can also add an email address if you don't want to wait for the results to load, and we'll send you an email with your results attached. So I'm going to submit these, and you can see that the threshold is listed at the top, the input sequences, and the trim sequences. So you can see that this one was trimmed of the C and F to get down to the actual CDR3 sequence. And then we get our results. So on the far left are the input sequences. Then we have the matching sequences. Those are the ones from the IDB that are found to match the input. There are links to receptor IDs where you can find more information. So if I click on this one, it'll take me to the IDB page where I can figure out exactly where this came from. I can find the publication that it's from, the assay, and just all that information is all connected. If I go back, the score here is listed as well, so you can see that there are some perfect matches which get a score of 1. Others fall between the threshold of 0.97 and 1. The epitopes are listed as well. The source organism for the epitope is here, as well as the antigen from which you get the epitope. And I think that's about it. There's an option to download your results as usual. You can also search. So if you wanted to just look at the perfect matches, you could just search for 1.0. That would lead you directly to the sequences that have been characterized before. And yeah, I think that's all I've got for the demonstration. So I'll just quickly switch back and just wrap up here. So we think that TCRMatch is an efficient way to find similar TCR sequences that have been curated in the IDB with known specificity. If you're in a situation where you have TCR repertoire data and you don't know what the epitope is, or you have a few epitopes that you think might be involved, this will help you sort that out. You could use it in tandem with TCR clustering tools such as glyph2 to determine likely epitopes for TCR repertoire sequences. One of the big advantages I think of TCRMatch is that it's not trained on specific epitopes, which makes it unbiased and generalizable, as opposed to some of the machine learning methods out there that are obviously trained on specific data sets, and as a result, they're subject to how good and how broad that data set is. In our case, we're doing something very simple, but something very useful in just looking for matches within the IDB. And we think TCRMatch as an algorithm does a good job of finding those similar sequences. So in the future, I think we'll continue to improve as we add more data. We're considering improvements we can make, such as replacing BLOSSOM62 with a CDR3 beta derived matrix. And we're also going to be evaluating whether we can improve performance if we integrate CDR3 alpha sequences, CDRs 1 and 2, as well as MHC information to improve our predictions. So with that, I want to thank everyone that was involved with this. It's certainly been a team effort, and Austin in particular has been a huge part of making this tool a success. So stay tuned. We'll hope to get that paper published in the coming months, and hopefully it'll be in the IDB tools site very soon. So with that, thank you.
Awesome. Thank you so much, Will. It's excellent to see the new work coming. And as I mentioned to the attendees, you're getting a behind the scenes look at this. So I hope you enjoyed that. And also thank you to Austin. That was a great shout out. Austin presented earlier, has been collaborating really closely with Will, which is awesome.